Okay, good afternoon, everyone. I am not Sarah Booth. I'm Roger Fielding. I'm the Associate Center Director at the Jean Meyer USDA Human Nutrition Research Center on Aging. And it's my pleasure to welcome you to the HNRCA Monday Seminar Series. It's my distinct pleasure to introduce Dr. Paul Jacques, who is our seminar speaker today. Paul is a senior scientist and team leader of the Nutritional Epidemiology team at the HNRCA. His research interests include B vitamins, whole grains, flavonoids, hydration, diet patterns, and diet quality, and their relation to the maintenance of metabolic, cognitive, and physical function with aging. Paul has had a leading role in the implementation, design, and assessment of nutritional intake as part of the Framingham Heart Study, and he's really played a pivotal role in all of the dietary work that's come out of that epic study over the last many years. Paul's published more than 300 peer-reviewed manuscripts of original research over his career and has made many important discoveries in the field of nutritional epidemiology. Among his most significant research accomplishments has been the observation that institution and mandated folic acid fortification in the United States had a profound impact on folate status of older Americans, including possible adverse health effects of high levels of folic acid consumption. He demonstrated that fortification increased circulating unmetabolized folic acid concentrations and that the presence of detectable circulating unmetabolized folate among older Americans with inadequate vitamin B12 status is related to lower cognitive test scores. Paul currently holds the rank of professor at the Friedman School of Nutrition Science and Policy at Tufts, and he's given over 100 invited presentations across the world and has served on numerous grant panel reviews. He is a member of the Society for Epidemiologic Research and American Society for Nutrition and is a fellow of the American College of Nutrition. So without further ado, it's my pleasure to introduce Dr. Paul Jacques. The title of his presentation today will be Nutrition and the Risk of Alzheimer's Disease. Dr. Jacques.
Thank you, Roger, for that wonderful introduction. Let me get my slides here. Okay, and good afternoon to everybody. Today, I'm going to focus my presentation on the epidemiologic evidence relating diet to risk of age-related cognitive decline in Alzheimer's disease. This is just an outline of what I'll discuss. I'm going to give a brief background on research relating to diet and cognitive function, highlighting some of the important and seminal contributions made by investigators here at the USDA and the Nutrition Research Center on Aging at Tufts. I will then summarize some of the epidemiologic evidence that diet is a key modifiable determinant of Alzheimer's disease. I will first present evidence relating healthy dietary patterns to Alzheimer's disease. Then I'll focus briefly on recent work that we've done examining the relationship between flavonoids and Alzheimer's disease. And I'll finish by discussing an ongoing study examining the potential role of B vitamins in Alzheimer's disease risk. Unfortunately, the COVID-19 epidemic has largely put that project on hold as we await the reopening of our core labs for analysis of the biomarkers of B vitamin status. So I will focus on some of the evidence largely developed here at the HNRC by Jacques scientists in my program, the Nutritional Epidemiology program, that led us to propose this project to the NIH and led the NIA to fund it. So I'll start with a very brief background on nutrition and age-related cognitive function in humans. There was no shortage of research on nutrition and cognitive function in the early and mid 20th century. Much of that work was related to nutrition and cognitive development in children. Research on nutrition and cognitive function in adults was almost entirely related to vitamin deficiencies and alcohol abuse. There was also some work examining the nutritional status of senile patients, but not from the perspective of nutrition relating to the development of cognitive decline, but rather the impact of cognitive decline on the nutritional status. From an epidemiologic perspective, though, the early 1980s was somewhat of a watershed moment for the study of nutrition and age-related cognitive function. In 1980, Dr. Phil Gary started enrolling a cohort of approximately 300 healthy individuals aged 60 and older from the Albuquerque, New Mexico area. He did this for a longitudinal study of aging. And in 1993, he published a paper from this cohort titled Association Between Nutrition Status and Cognitive Function in a Healthy Elderly Population. And this slide is a figure from that paper that relates the scores from a classic test of executive function in those with the lowest and highest biomarker levels of various vitamins. You can see from this figure that the healthy older participants with the lowest levels of vitamin C, riboflavin, and vitamin B12 scored worse. That is, they had more errors on this test of executive function than those with the highest vitamin levels. And I think one important aspect of this was the paper itself, but also the accompanying editorial for this paper, because I believe it helped to shed light on the state of the science of nutrition and cognitive function in aging individuals at that time in 1980, and also the beliefs regarding this association during that time period. And so just let me quote from this editorial. Oops, sorry about that. The person writing the editorial stated that classic vitamin deficiency diseases such as pellagra, Wernicke syndrome, scurvy, and pernicious anemia all manifest cognitive dysfunction and even frank dementia as part of their presentations. Is it not then possible that subclinical nutrient deficiencies described in both institutionalized and community dwelling elderly may impair cognitive function in later life? So this is really, I think, the first formal recognition that subclinical deficiency might be important, and these various vitamins might be important for age-related cognitive decline. Now let me skip ahead about a decade to highlight some of the groundbreaking work done at the HNRCA on nutrition and age-related cognitive function. I first want to highlight the work of Barbara Shugart-Hale and Jim Joseph and colleagues. I highlight this work not only to show the key contributions that the HNRCA has made to the role of nutrition and cognitive function on aging individuals, but also because it is a foundation for some of the work that I'll present later on flavonoids and Alzheimer's disease. And it was also critical, this work was also critical to the development of one of the healthy dietary patterns that I'll talk about today that was developed solely for the, to examine the maintenance of cognitive health in older age individuals. And this was called the MIND diet. I won't go through the work here, Barbara's work in detail given the time constraints, but I want to point out that this slide is only a representative sampling of Barbara and Jim's work on cognitive function. It represents their early recognition of the role of polyphenols in maintaining cognitive health. It also represents both their animal work and their later clinical work in humans. So to conclude my background, I'd also like to briefly highlight more groundbreaking work done at the HNRC relating nutrition and cognitive health at older ages. Again, in this slide, I have included selected papers highlighting the contributions of Dr. Elizabeth Johnson and colleagues to our understanding of the relationship between lutein and cognitive function. This list starts with Dr. Johnson's trial showing benefits of lutein and DHA on cognitive function of older women. It also includes her critical work showing that brain concentrations of lutein are related to cognitive function in the Georgia centenarian study participants. And finally, her work relating dietary lutein to brain lutein concentrations in primates and humans. As with Barbara's work, I highlight Liz's work not only to show the contributions of the HNRCA to our understanding of the role of nutrition and maintenance of cognitive health, but also because, as I will show in a few minutes, this work also helped contribute to the development of the mind diet. Before I go on to present the relationship between the different dietary patterns and Alzheimer's disease, I'd like to spend a minute just to provide a short introduction to dietary patterns. For much of the 20th century, the primary focus of nutrition science that was examining the relationship between diet and disease was on individual foods or food groups, individual foods or food groups, and nutrients or non-nutrient constituents. This approach, this reductionist approach, helps us to understand the underpinnings and the mechanisms relating nutrition and health. In the past 20 years, this approach has been complemented by the use of dietary pattern methodology. So why dietary patterns? Well, from a research perspective, dietary patterns present an advantage for understanding the relationship between diet and health, because they overcome many of the limitations of this reductionist approach, particularly for observational studies. For example, one important limitation of a focus on a single food or nutrient is the highly interrelated nature of their exposures. People who consume diets that are higher in fruits and vegetables are going to have higher intakes of many vitamins. They're going to have higher intakes of potassium and fiber and polyphenols. And they're also going to have lower intakes of saturated fat, sodium, and added sugar. So these types of patterns that we see in diets make it difficult to separate out the effects of a specific nutrient or food. Another important use or application for the dietary patterns is really to be able to look at the interactions and of these different nutrients in foods. A focus on a single nutrient or food does not capture these potential additive or interactive effects of the different foods and nutrients. While the impact of a single nutrient on a health outcome might be small and unmeasurable, the cumulative and synergistic effects of multiple nutrients and non-nutrient constituents as part of a diet pattern might result in a much larger and measurable impact. There are many different types of dietary patterns. Today, I will focus on what are known as criterion-based patterns. All this means is that there are specific guidelines that define the pattern. For example, the Healthy Eating Index, which I'm sure many of you are familiar with, is a criterion-based pattern using the recommendations from the dietary guidelines for Americans. There are two types of criterion-based patterns: the traditional patterns and patterns based on dietary guidance or recommendations such as the HEI. Today, I will consider evidence from one traditional dietary pattern, the Mediterranean diet, and two patterns based on dietary guidance, the DASH diet and the MIND diet. Let me quickly show you the reminder of the major characteristics of the Mediterranean pattern. As you can see in this slide, the Mediterranean pattern is largely a plant and fish-based diet with a focus on use of olive oil. And it also restricts intake of red meats and sweets. So this is the earliest study to examine the relationship between a Mediterranean diet and incidence of Alzheimer's disease. It included over 2,000 community-based non-demented individuals from New York City, from the New York area, who were prospectively evaluated every year and a half for an average of about 4 years of follow-up. Better adherence to the Mediterranean diet pattern, as shown in this slide, was associated with a 40% lower risk of incident Alzheimer's disease after adjusting for several personal, lifestyle, and clinical characteristics. This observation was replicated in 6 of the 10 other studies that examined this association. One of those studies is shown in this slide was a more recent study by Martha Claire Morris and colleagues. They conducted this study among 1,000 participants of the Rush Memory and Aging Project. This is a study of volunteers living in retirement communities and senior public health units in the Chicago area. Participants were followed for an average of about 4.5 years. And as you can see, the study showed a greater than 50% reduction in the incidence of Alzheimer's disease among those adhering most closely to a Mediterranean diet pattern. So this observational work on Mediterranean diet and Alzheimer's disease is supported by clinical findings from the Predimed trial. At baseline and at the end of the study, as part of the Predimed trial, people were administered neurocognitive tests. And as shown in this slide, participants on the Mediterranean diet arms maintained or improved cognitive function on multiple tests over the course of the trial. The controls, on the other hand, that had the cognitive function worsened on average on cognitive tests for memory, frontal lobe cognition, which is a composite of tests measuring attention, cognitive flexibility, and working memory, and also on global cognitive function. So the second diet pattern that I want to consider today is the DASH diet pattern. There's actually very limited data relating the DASH diet to cognitive function, and only one study by Dr. Morris and colleagues relating the DASH diet to Alzheimer's disease. Given this very limited and equivocal evidence, I present this pattern only because of the MIND diet, which we'll discuss next. The MIND diet, according to its developers, draws its criteria from the Mediterranean and DASH diet patterns. However, I will show you later that the MIND diet is essentially a slightly modified Mediterranean diet. And apart from the common aspects of the Mediterranean DASH diet, I see no unique contributions from the DASH diet to the MIND diet. But nevertheless, because of the MIND, because of the involvement and theory of the MIND diet, of the DASH diet and development of the MIND diet, I will present the results. So how does the DASH diet differ from the Mediterranean diet? Well, there's two important differences. One is the type and amount of fat. The DASH diet pattern does not emphasize olive oil, but restricts both saturated and total fat. So the DASH diet is a low-fat diet pattern, unlike the Mediterranean diet. Also, the Mediterranean diet is mainly a plant and fish-based diet, while the DASH diet is mainly a plant and low-fat dairy-based diet. As I just mentioned, there's only one study, which was conducted by Dr. Martha Claire Morris and colleagues, that examined the relationship between adherence to the DASH diet and incidence of Alzheimer's disease. And as shown in this slide, they observed a 40% reduction in the incidence of Alzheimer's disease in those who were more adherent to the DASH diet pattern. Now, let's talk a little bit about the MIND diet. So the same investigators, Dr. Morris and her colleagues, created a new diet pattern, and they specifically designed this pattern for protection of the brain. They call this pattern the Mediterranean DASH diet intervention for neurodegenerative delay, or more simply, the MIND diet. So this diet was proposed as a mix of the Mediterranean and DASH diets, but with modifications based on evidence relating, pre-existing evidence relating diet to cognitive function. So the investigators believed that the evidence relating all fruit consumption and cognitive decline was weak, but the evidence relating berries and protection against cognitive decline was quite strong. And again, this is this is largely based on the work done by Barbara and her colleagues here at the HNRCA. They also felt that the strongest evidence relating vegetables to cognitive decline was seen with green leafy vegetables. This evidence was derived in part from the work that Dr. Morris did in collaboration with HNRC investigators Bess Dawson Hughes and Sarah Booth in examining nutrients and bioactives in green leafy vegetables in relation to cognitive decline. And also, this is based in part on the work I previously mentioned from Dr. Johnson on lutein and cognition. So let's compare this resulting MIND diet to the Mediterranean DASH diets. If we do, we can clearly see, I think, that it is heavily influenced by the Mediterranean diet. The only major differences, which I've highlighted here, are the focus on berries and green leafy vegetables. And there's one other a difference in that the MIND diet elevates the poultry from the moderate consumed category to the highly consumed category. I'm not sure, I could never find any rationale for the basis of this elevation. So these three differences also apply to the DASH diet, the differences in the poultry, in the berries, in the vegetables. But apart from the similarities with the Mediterranean diet, I just don't see how the DASH diet has influenced the MIND diet, apart from the fact that it's part of the name of the diet. So, as shown in this slide, Dr. Morris and colleagues first demonstrated that adherence to the MIND diet was associated was positively associated with global cognitive function and function in 5 cognitive domains. And this again was done in the Rush Memory and Aging Project. They also examined the relationship between the MIND diet and incidence of Alzheimer's disease in the same population. And they demonstrated a greater than 50% reduction in incidence of Alzheimer's disease among the participants with the highest adherence to the MIND diet pattern compared to those with the least adherence to this pattern. So I'd like to conclude with a brief, this brief presentation of healthy diet patterns on Alzheimer's disease with a brief summary and some interpretation of the evidence. We see that healthy diets, even with some variation, are strongly associated with Alzheimer's disease incidence. Variation is largely related to animal protein sources that are encouraged and fat sources and amounts. This suggests that the risk of Alzheimer's disease is being affected by the common elements of these 3 diet patterns, which include the focus on plant foods and limited red meat and sweetened foods. Also, given the strength of these associations, they are unlikely the result of confounding, a point I will address in greater detail later. However, given the relatively short follow-up times for many of these studies relating these diet patterns to dementia, we cannot rule out the possibility of what some call reverse causation. That is, the outcome affecting the exposure. In the case of Alzheimer's disease, it is highly likely that declining cognitive function prior to the development of Alzheimer's, to the diagnosis, could affect not only the dietary intake, but also the reporting of that intake. Again, I will address this issue in greater detail a bit later, but this is a bias that we cannot entirely rule out in these studies. Even though they are prospective studies, they're relatively short follow-up periods just don't allow us to dismiss this type of bias. Next, I'd like to focus a little more on some of the potential bioactive constituents of these dietary patterns. Based on the earlier work from Barbara on berries and polyphenols and the fact that the Mediterranean mind diet patterns are good sources of flavonoids, I initiated a project to examine the association between usual flavonoid consumption and risk of incident dementia and related dementias. Incident Alzheimer's disease and related dementias. This work was done largely by Dr. Ezra Shishtar, formerly of the HNRCA and the Friedman School, in collaboration with Jeff Blumberg from the Friedman School and Rhoda Au from Boston University. This slide here is my obligatory flavonoid slide showing the 6 different classes of flavonoids and the flavonoid polymers. These polymers are derived from the flavin thiols, as this is depicted in this slide. For our work, we focused on all classes of flavonoids, except for the isoflavones, because their intake in the US still remains fairly low. This slide shows the common sources of the different flavonoid classes. For my presentation today, I will be focusing on flavanols, which are derived largely from apples, teas, yellow onions and scallions, kale, broccoli, and berries. The flavonoid polymers, which are derived largely from teas, particularly black and oolong, chocolate, and apples, berries, red grapes, and red wine. And the anthocyanins derive largely from berries, red, blue, and purple, and red grapes and red wine. To examine this relationship between flavonoid intake and Alzheimer's disease, we used data from the Framingham Heart Study Offspring Cohort. We studied 2,801 participants with a mean baseline age of 59 years who were free of dementia at baseline. We captured flavonoid intake from 5 clinical examinations over a 20-year period. And in our analysis, we updated the flavonoid intake at each exam so that the exposure represented the cumulative average intake across the 20-year follow-up. During follow-up, we identified 193 total cases of Alzheimer's disease and related dementia, of which 158 were Alzheimer's disease. So after multivariate adjustments, we observed that individuals with the highest intakes of flavanols had a 49% reduction in total incidence of Alzheimer's disease and related dementia relative to those with the lowest intakes. We also saw a significant 47% reduction in risk of Alzheimer's disease and related dementia for participants with the highest flavonoid polymer intakes relative to those with the lowest intakes. And finally, for all of us berry lovers, we observed a remarkable nearly 75% reduction in the incidence of Alzheimer's disease and related dementia for participants with the highest anthocyanin intakes relative to those with the lowest intakes. These observations, we also looked at these observations in Alzheimer's disease cases alone and saw very similar associations. So we did not observe any associations between flavanone and the flavin thiols intakes with either Alzheimer's disease and related dementias or Alzheimer's disease alone. And total flavonoids were significantly related to Alzheimer's disease and related dementia, and a sensitivity analysis we did using a 10-year period between Alzheimer's disease diagnosis and the updated flavonoid intake. So this study was unique for a number of reasons, including the length of follow-up and the number of dietary assessments performed during the follow-up. Typically, such studies have an average follow-up of about 5 years and dietary data based on a single baseline assessment. We had an average of 20 years with 5 measures of diet. Also, these types of studies tend to include participants who are older at baseline, typically in their 70s and 80s, and already at risk of Alzheimer's disease. Our participants on average were 59 years old at baseline. This allowed us to capture dietary intakes in midlife prior to being at risk of AD. This is important for 2 reasons. First, it speaks to the possibility that diet in midlife can still influence risk of Alzheimer's disease, but it also allows us to account for an important source of potential bias in all studies of nutrition and age-related cognitive dysfunction, in this issue of reverse causality, which I'll speak to in a minute. So let me address all the issues of potential sources of bias in this study. So first, we wanted to assure ourselves that the findings were not a consequence of confounding. So we adjusted for several clinical and lifestyle factors, as all these studies do, but also we included adjustment for overall diet quality. This adjustment for diet quality is critical when we're focusing on individual foods and nutrients such as flavonoids, given the interrelationship between dietary components that I had described earlier. However, one can never rule out the potential for residual confounding. That is, confounding associated with unmeasured or inadequately measured confounding factors. Residual confounding is often used to dismiss observational findings such as ours. The argument is often made that residual confounding by lifestyle factors can readily account for any associations seen with diet. That may be a reasonable argument if we observed a 5 or 10% reduction in incidence, or perhaps even a 20% reduction. However, for residual confounding to be responsible for a 50 to 75% reduction in incidence of Alzheimer's disease, the unknown or poorly controlled confounder would have to be nearly 100% correlated with flavonoid intakes and have a relative risk for Alzheimer's disease equal to or even greater than that which we observed with the flavanol intakes. Thus, for our results to be a consequence of residual confounding, we need a factor that has a relative risk for Alzheimer's disease of greater than 2-fold or less than 0.5 and nearly perfectly correlated either positively or negatively with flavonoid intake. I'm not aware of any such factors that fit these criteria, so I would dismiss the potential residual confounding alone being responsible for our findings. However, as I mentioned earlier, the most likely bias in any study of diet and cognitive dysfunction would not be confounding, but rather would be reverse causation. As I noted earlier, in the case of Alzheimer's disease, it is highly likely that declining cognitive function could affect not only dietary intake, but also the reporting of that intake. The potential bias is not often considered in studies in nutrition and Alzheimer's disease. This was a major concern for us when we were designing the study. So we designed it to include a minimum 5-year period between the diagnosis of dementia and the last update of flavonoid intake. We also performed sensitivity analyses to assure ourselves that the 5-year period between the last cumulative update of flavonoid intake and dementia diagnosis was sufficient. We did that by extending that time period to over 10 years. When we did that, we saw that the associations between flavonoid intakes and Alzheimer's disease incidence was either unaffected or even tended to strengthen. So again, we are confident that our observations are not a consequence of either confounding or reverse causation. So let me now transition to B vitamins and cognitive function. The notion that B vitamins might affect cognitive function has been with us for some time based on the the cognitive dysfunction seen in deficient patients. This is particularly true for B12. But relatively little work has been done to specifically look at the relationship between inadequate B12 and folate levels and age-related cognitive decline. And the existing literature is equivocal. Our first study to examine the relationship between B vitamins and Alzheimer's disease relied on plasma homocysteine levels. This was a marker, we use this as a marker of inadequate levels of folate in B12. We examined this association in the Framingham heart study cohort as shown in this slide. And we observed that participants with elevated homocysteine levels had approximately a twofold increased risk of incident dementia compared with those with normal homocysteine levels. And the findings were similar when we looked specifically at Alzheimer's disease cases. The story of B vitamins and cognitive function quickly got a bit more complicated. In 1996, the FDA mandated fortification of enriched cereal grain products with folic acid. And this dramatically changed the folate status of Americans. Roger, in the introduction, had noted some of the work we had done looking at the impacts of fortification. And we were the first to actually show the impact of fortification on folate status. We used longitudinal data on folate status from before and after fortification that we had collected in the Framingham offspring cohort. And as you can see in this slide, prior to fortification, in the white line, about 25% of the Framingham population had levels below 3 nanogram per mil for plasma folate, which would be considered to be inadequate levels. After fortification, you can see the dramatic change to the point where almost no one had inadequate folate status. And this is among non-supplement users. Also, one thing to note is prior to fortification among non-supplement users and even among supplement users, the probability or the prevalence of levels of folate over 20 were really uncommon. Whereas after fortification, we see those levels even among non-supplement users and particularly among supplement users. So why is this important? Well, there's a long-standing hypothesis that high folic acid intake causes exacerbation of neurologic symptoms associated with inadequate vitamin B12 levels. And unfortunately, inadequate vitamin B12 status is too common among older adults. This hypothesis is based on observations from the mid-1950s through the 1960s that pernicious anemia patients who were prescribed this newly synthesized folic acid without B12 treatment appeared to have worsening of their neurological symptoms. And now, with the extraordinary and unprecedented levels of folic acid in our diets, we have the opportunity to address this old hypothesis. And this story got a bit more interesting as well, based on work we did with Dr. Jacques examining the relationship between higher post-fortification folate levels and markers of B12 status. This slide is based on post-fortification blood levels of folate and B12 and a functional marker of B12 status, methylmalonic acid. Higher methylmalonic acid levels indicate a greater functional or intercellular B12 deficiency. In the left panel of this slide, you can see that elevated folate levels do not affect methylmalonic acid of individuals with normal B12 levels. However, among those individuals with with inadequate B12 levels, these elevated folate levels dramatically increase the amount of methylmalonic acid. This indicates that the high folate levels may be worsening the actual B12 deficiency among those who already have inadequate B12 levels. And these findings were supported by the work of Dr. Joshua Miller, an HNRCA alum, who also showed in a salsa population that methylmalonic acid levels were elevated among individuals with, as you can see on the right side, among individuals who had both low B12 and elevated folate levels. So there were two critical questions that we set out to address. First, is low B12 status associated with a higher risk of age-related cognitive decline and Alzheimer's disease? And if so, is that relationship affected by elevated folate levels? So again, using the NHANES participants, Dr. Martha S. Morris, formerly from the HNRCA Nutrition and Epidemiology program, and not to be confused with Dr. Martha Claire Morris from Rush University, which happened all the time. She examined the cross-sectional association between folate, vitamin B12, and cognitive impairment. Cognitive impairment was based on a very low digit simple substitution test score. As can be seen in this slide, relative to those with normal B12 and normal folate levels, those with normal B12 and elevated folate levels had a significant reduction significantly lower cognitive impairment. And that's not unexpected because there is evidence that higher folate levels are associated with better cognitive function. However, if we look at those individuals who have low B12 levels and normal folate levels, we see about a 70% increased risk of cognitive impairment, which is consistent with an effect of B12 alone on cognitive function. However, if we look at those individuals who have inadequate B12 and elevated folate levels, we see a 5-fold risk of cognitive impairment. So these findings clearly support the hypothesis of an interaction between elevated folate and inadequate B12 levels, and that this interaction is manifest by changes in cognitive function in older adults. Now, this study I just showed was cross-sectional, which has some limitations. So Dr. Morris went on to examine this interaction in an older population from the Framingham heart study, using longitudinal data, looking at change in the mini mental state exam test scores over an 8-year period. Interestingly, this study was done pre-fortification. So it was done prior to the elevated levels of at least a high prevalence of elevated levels of folic acid in the diets. But in spite of that, Dr. Morris again observed this interaction. As you can see, again in this slide, individuals at normal folate levels, individuals with inadequate B12 tended to have a greater loss in the change in the mini mental state examination score over the 8 years. However, you can see the dramatic drop a dramatic change, dramatic loss of cognitive function among those individuals who had elevated folate or elevated folate levels. So these observations, as well as many others, prompted us to propose a study of vitamin B12 and folate status and healthy brain aging to the National Institute for Aging, which was successfully funded. The study is being conducted in the Framingham Heart Study offspring cohort. The aims of this project are to examine longitudinal relationships between a novel and sensitive and specific measure of vitamin B12 status that we were employing, and incidence of Alzheimer's disease and related dementias, changes in neuropsychological test scores, and changes in brain structure based on MRI. We have baseline data for nearly 3,000 participants to assess the relationship between B vitamins and incident dementia, with almost 30 years of follow-up. We will also assess the relationship between B vitamins and changes in neuropsychological tests and brain structure in about 2,000 participants measured at 3 time points over 14 years of follow-up. And for all of these aims, we will assess the impact of elevated folate levels on these associations. This project is, as I mentioned earlier, this project is currently on hold awaiting analyses of the B vitamin biomarkers for the follow-up exams. That analysis has been delayed due to the COVID-19 shutdown of our core nutrition evaluation lab. But we hope to get this study back on track and have some preliminary data by next summer. So just let me finish by acknowledging all those who've contributed to this work. First, I'd like to acknowledge Ezra Shishkar, who did the lion's share of the work on the Flavonoid project and indirectly provided a great deal of assistance on the B12 project through her cleaning and providing us with a better understanding of the very complex longitudinal dementia, neuropsych, and MRI data sets. Gail Rogers, our program statistician, supported Ezra on the Flavonoid project and is responsible for the statistical analyses on the B vitamin project. Carol Livingston is the project manager for the B vitamin project and for everything else we do. And Catherine Barger is the senior statistician on the B vitamin project, advising Gail and me on the best statistical approaches for these complex longitudinal analyses. I would like to acknowledge all of the incredible input and new ideas and energy that Jacques has put into all of our B vitamin collaborations, both past and ongoing. Prior to his retirement, Jacques was a multi-PI on the B12 grant and remains a consultant and a key advisor on that study. I would also like to acknowledge my current multi-PI on the project, Joshua Miller from Rutgers University, and Jeff Blumberg from the Friedman School and Rhoda Au from BU were both co-investigators on the Flavonoid project, and Rhoda is a co-investigator on the B12 project. And finally, as I lose my voice here, last but not least, Bruce Fischel, a Harvard neuroscientist, is also a co-investigator on the B12 project. With that, I thank you for your attention, and I'd be happy to take questions.
Thank you so much, Paul, for a really excellent seminar. The questions are already coming in via the Q&A, and I would just remind people, if you do have questions, to please submit them through the Q&A function in Zoom. And I'll start with the first question from Alan Taylor. Alan asks, the effects of fish oil on diet patterns and dementia. Also, the AOR cohort had only a 1% incidence of Alzheimer's disease. How does that compare with incidence of AD in the general public?
So it was a 1% incidence, did you say?
I think that's what he said in his question.
Yeah, 1%. Well, it's hard to say whether that's what age group and whether that's cumulative. So it's hard to, you have to put that in context, but that's low. I mean, if you're looking at the prevalence of dementia at older ages, it can become quite substantial. So once you get into your age, and also the age group being studied study really matters, because the prevalence is very low until they start getting into your early 80s, mid 80s.
Okay, Nirupa Mathan asks, in the flavonoid and AD study, you mentioned that adjustment for overall diet quality was conducted. Can you expand on how this was done? Did you assign a diet score for each individual?
Yes. There's many different ways of doing that, any kind of diet quality score. We've developed one internally, which is referred to as the Dietary Guidelines Adherence Index, which is similar in many ways to the Healthy Eating Index. And basically, it can account for the for overall diet quality. So yes, so we basically assign a score to each individual.
Okay, and we have a question from one of our national program leaders at the USDA, Dave Klorfeld. Do you have data on ApoE polymorphisms in the Framingham cohort?
We do, and we actually, for all of our dementia work, we have adjusted for that. We've seen, we actually looked for interactions as well with diet, and we didn't see any interaction with the ApoE genotype.
Okay, Iris Bowles asks, should we consider taking B12 or adding it to our diet through foods like soy milk, or is it too soon to say?
Well, I mean, you have to, as far as dementia or Alzheimer's disease, I would probably say it's too soon to say. Let's finish our study first. But I mean, there is a recommendation that older individuals take multivitamin supplements, including B12, because of the higher prevalence of B12, inadequate B12 levels in older individuals. So whether it is through some kind of fortification of food or through supplements, older individuals should be seeking out sources of B12 to make sure they have adequate B12 levels.
And Zhendong Wang asks, could you comment on potential effects of high refined carbohydrates, sugar diets to the risk of pre-AD cognitive decline, or pre-AD or AD related cognitive decline?
Yeah. Again, I can't specifically speak to that. Again, it gets back to the diet pattern and the fact that the diet patterns that have lower intakes of refined carbohydrates and sugars tend to show benefits. So that might be part of the overall diet pattern associated with this. But the again, there's probably so many different factors contributing to these large strong associations we see with these healthy dietary patterns. And I would suspect that refined grains may play a role there, but there hasn't been a lot of work done looking at refined grains and sugars with respect to dementia yet.
We have a question from an anonymous attendee asking about the relation between fish intake and food patterns as well as with AD. So I guess the question is about maybe just about fish intake and AD risk.
Yeah, well, there's a lot of information on a DHA and dementia and Alzheimer's disease, and much of that is supportive of benefits from fish oils and protection from fish oils for AD incidence. The Mediterranean diet is typically a high fish diet, but again, we're also seeing benefits or beneficial associations with diets that don't necessarily include high fish, such as the MIND, well, the MIND diet does, but the DASH diet. So it's not in terms of the, again, I'm a proponent of dietary patterns for many reasons, but I think that diets that are high in fish have been we can clearly say that diets that high in fish have been demonstrated to be associated with lower risk of Alzheimer's disease, and as well diets that are diets high in DHA have been specifically associated with lower risk of dementia. So I mean, I think fish probably plays an important role. And there's reason to believe, based on a mechanism as well, that DHA is playing an important role.
Okay. Sarah Booth says, Paul, excellent seminar. So congratulations. Resilience is an important concept in dementia. What are your thoughts around dietary patterns and resilience to other factors that may cause AD?
That's a good question. I haven't thought about it much, to be honest with you. So I really can't comment on that, but I think it's a very good question. You know, again, you're getting back to the issue of how that might relate to the diet patterns as well. So unfortunately, I can't give a good answer to that because I really haven't given it much thought.
Okay. A question from Alice Lichtenstein. Can you rule out an interaction between healthy dietary patterns and optimal physical activity patterns?
No, it's an easy question. No, we can't. You know, again, that both physical activity and diet seem to be important contributors. But I mean, people ruling out is sometimes hard because there have been studies that have actually looked at interactions between diets and physical activity. But a negative interaction doesn't mean much, given that you need a larger samples that are more power to detect interactions. So I don't think we can rule them out, but I don't think there's much evidence to suggest the presence of an interaction between physical activity and diet with respect to Alzheimer's disease incidence.
Because there's just not a large enough study that would give you the power to tease it out, or?
Yeah, I don't. Yeah, I mean, it's hard. I mean, even we have 3,000 participants in our study, in our Framingham study. What we're lacking is a really good measure of physical activity in Framingham. But even with 3,000, it might be hard to really see an interaction with physical activity. And again, it depends on when you're looking. One of the hard things we have is really trying to figure out what's important. Is physical activity and diet important in midlife, or is it important at age 75? And you know, again, the difference in physical activity between midlife and 75 is going to be, for most people, is going to be substantially different. So it's not only the ability to measure it and the power, the sample size needed to look at it, but also the timing.
Yeah, is it a trajectory? Is it some absolute minimum level you have to maintain, or yeah?
Yeah, I mean, that's what we've tried to do with the diet. Is we're really looking at trajectory of diet over time, starting at midlife with the Framingham midlife and looking into older ages.
So I'd like to ask lots of questions, but I'm trying to keep to the audience question. Paul, was egg intake adjusted for in the MIND diet when poultry was elevated to the highly recommended category?
No, but I don't think poultry would be, eggs typically would be considered a separate food from poultry. So that, I don't think that was included. I mean, but egg intake is interesting because eggs are high in lutein. And as I mentioned, lutein is believed to be beneficial with respect to cognition as well.
So we are just about at 1 o'clock, and I'm not going to be able to get to all the questions people asked, but there is a good sort of last final question. And it comes again from another anonymous attendee, but it does ask, what would be your bottom line layperson advice in diet for we who are elders listening to your important presentation?
My advice, I think my advice would be, for an elder, I would say take a B12 supplement, first of all, but my advice would be, look at the diet like a Mediterranean diet pattern. I mean, you really want a MIND diet. These are really, they're plant-based largely with limited animal protein sources. And in the case of the Mediterranean diet, the focus is really on fish with moderate intakes of poultry and limited intakes of red meat. And in terms of the Mediterranean, poultry gets up there with the fish. But it's largely, if you look at the foods, it's largely plant-based, avoiding sweets and red meats and processed meats. And I think that's, that type of diet, I think the Mediterranean is a good pattern to try to emulate.
Well, that's a great way to sum up what was really an excellent seminar and question and answer session. I'd like to again thank Paul Jacques, our speaker today, for his presentation and his Q&A with us. And just remind everybody that we do have a seminar next Monday at the same time, and it will be Dr. Tong Zhang from the neuroscience group. So we look forward to seeing you all next Monday as well for our continuation of our seminar series. Thanks again, Paul, and thank you to everyone that listened in. Okay, great job, Paul. That was really excellent.
Thank you. Great.